Welcome everybody to the session on immunotherapy and the microenvironment. We know that we have a competing, equally exciting session going on at the same time. That's why we have assembled a stellar panel of speakers, because we want all of you guys to listen to us. And we have 4 speakers that will talk about the exploding new information that are coming up in the tumor microenvironment and immunotherapies of both central and peripheral nervous system tumors. So it's my great pleasure to start introducing you. My co-chair will take over after the presentation, Dr. David Gutman from Washington University, who will give the introductory talk on immunology of the brain tumor microenvironment. Thank you.
Thank you, Antonio. So I'd like to spend the next few minutes talking about some of the insights that our lab has derived over the last 15 some odd years, trying to dissect the non-neoplastic tumor microenvironment. As everybody in this audience knows, NF1 low-grade gliomas are ecosystems. So they're not only composed of neoplastic cells, like bees would be composed of queen bees that are the reproductive species, but also workers and drones, non-neoplastic cells that are essential for tumor initiation and growth. In this regard, when you analyze the low-grade brain tumors in kids with NF1, as many as half of the cells in the tumors are non-neoplastic cells. This has been revealed to us in studying murine models of NF1 by single cell RNA sequencing, by bulk RNA-seq deconvolution, and by immunohistochemistry on both human and mouse tumors in this respect. And it's shown us that in the non-neoplastic compartment, there are predominantly 3 cell types to be concerned about. There are monocytes, microglia, and macrophages, which compose the largest portion of these tumors, as many as 30 to 45%. And then a small proportion of cells that are of the T lymphocyte variety. In using a genetically engineered mouse model of NF1 optic glioma, in which every mouse is born with a germline inactivating mutation of the NF1 gene that's coupled with Cre-mediated somatic loss of NF1 in neural stem cells, nearly 100% of the mice develop optic gliomas. These tumors are characterized by architectural distortion and swelling of the prechiasmatic optic nerve and the chiasm, and are also evident by small animal MRI. To first demonstrate that these monocytes are really important for optic glioma formation and progression, we took initially a pharmacologic approach. And these are studies that were initiated by Winnie Pong when she was in the lab and followed up by Yuan Pan. By inactivating microglia using a crude inhibitor like minocycline, or depleting them using the Plexicon compound, or by blocking the signaling pathway that regulates microglia activation, all of these events lead to reduced tumor growth. In addition, if you make the microglia unable to respond to the chemotactic factor fractalkine, or CX3CL1, by mutating one copy of the fractalkine receptor in optic glioma mice, you can delay the formation of optic gliomas by nearly 2 months. Well, the question came, well, how do microglia regulate NF1 optic glioma growth? Do they do this by secreting a growth factor? And if so, what is that growth factor? And this was tackled by a graduate student, Anna Solga from Berlin, who came to spend time in the laboratory. And working with Elaine Martis at the McDonald Genome Institute, she performed RNA sequence analysis of microglia from mice that had an NF1 mutation but no tumors, as well as mice that contain microglia that had that same germline mutation but resided within the tumor cells. So comparing these populations of NF1 mutant microglia that only differ by the fact that one of them is within the tumor, Anna was able to identify CCL5, typically known as a chemokine, as a growth factor that was differentially expressed by the glioma-associated microglia. To prove that CCL5 was required for optic glioma cell growth in vitro and in vivo, Anna partnered with Yuan Pan in the lab to show first that CCL5 by itself was sufficient to increase glioma cell growth in vitro. And if you neutralize CCL5 using a neutralizing antibody, you can dramatically attenuate tumor growth in vivo. So both necessary and sufficient. And importantly, using a model system where we could grow stem-like cells from these optic gliomas in mice and transplant them into wild-type mice, Yuan was able to show that the same tumor glioma stem cells could form tumors in wild-type mice, but not mice lacking CCL5. Well, who are the monocytes in these murine NF1 optic gliomas? We've called them microglia, but are they microglia or macrophages? We believe that they are microglia for several reasons. One is by FACS, these are CD11b expressing CD45 low and fractalkine receptor positive cells. And by immunohistochemistry, they express the microglia specific marker TMEM119. So we'll be referring to these as microglia for the rest of the chat. But are these microglia a specialized population because they are induced in the context of an optic glioma to make CCL5, whereas they're not doing that in just a mouse with an NF1 mutation? And to do that, we decided to look to see how we might distinguish glioma-associated microglia from brain microglia. And this is work that was spearheaded initially by Olivia Cobb, a bioinformatician in the lab, who took advantage of published microglia data sets from a number of CNS diseases, ALS, autism, and brain tumors, to identify a single molecule, a cell surface protein, CD11A, that is expressed in glioma-associated microglia. The importance of this was furthered by Amanda Costa, a postdoc in the lab, who demonstrated that the microglia in these optic gliomas, regardless of which optic glioma strain we used, expressed CD11A. So it was a glioma-associated microglia marker. And it also is enriched in microglia that are expressed in low-grade human tumors, or pilocytic astrocytomas. But the question remained, is this population of CD11A positive microglia important for optic glioma growth? And to do this, Amanda took advantage of the fact that we had been using blocking antibodies against cell surface proteins for a couple of years now to silence their function. And so she first isolated microglia from mice that lack CD11A and showed that they had impaired CCL5 expression. So if you don't have CD11A, those microglia are not able to generate CCL5. And if you treat mice with a neutralizing antibody against CD11A to deplete that population, you not only reduce that population, but reduce the volumes of the tumors, the proliferation of the tumors, and the number of microglia in those tumors. So establishing that there is now a population of microglia that are unique to the tumors and are important for generating CCL5, the question then becomes, how is CCL5 expression induced in the microglia? And to do this, we were responding to what we thought at the time was an unusual request by reviewer number 3. When Yixin Chen in the lab submitted his paper on optic glioma stem cells forming glioma-like lesions in wild-type mice, the reviewer said, that's all fine and good, but the gold standard in the field is to put them into nude mice. So if you want your paper accepted, please transplant those optic glioma stem cells into athymic nude mice, which we did. And Yixin found that they formed no tumors whatsoever. If you look at the microglia from athymic nude mice, they have virtually undetectable levels of CCL5. So much like those CD11A knockouts, if you impair the ability of these T cells, of these microglia to get educated by the T cells, they also don't make CCL5. And then importantly, if we take these optic glioma stem cells that could not form tumors in nude mice, but gave them their own CCL5 by ectopic expression, they now can form tumors in the athymic nude mice. So the defect was the T cells educating the microglia to make CCL5. To demonstrate that T cells are actually important for optic glioma growth, Gary Guo, a graduate student in the laboratory, used a number of strategies to inhibit T cells. And in these experiments, he either blocked T cell brain entry using an anti-VLA4 antibody or blocked T cell function using an anti-CD3 antibody. And in all cases, that reduced the proliferation of the tumors to below or at wild type levels. But how do the T cells in these tumors educate the microglia? And to cut a very long story short, what Yuan Pan in the lab did was demonstrate that it was a paracrine factor that was made by activated T cells that stimulated microglia to make CCL5, and that paracrine factor was the cytokine CCL4, and that CCL4 made by these activated T cells was sufficient to program the microglia to make CCL5 and allow the tumors to grow. So we now know that the T cells in the context of the tumor make CCL4 that activates the microglia to make this important gliomagen, this glioma growth factor, CCL5, and that this operates through NF-kappa B signaling. Jit Chatterjee in the lab showed that the ability of the T cells to instruct microglia to make CCL5 is indeed controlled by NF-kappa B, because if he inhibits NF-kappa B signaling, he can reduce the ability of those microglia to respond to T cells and make CCL5. And then importantly, if he treats mice in vivo during the process of glioma evolution, he can block tumor proliferation 6 weeks after the cessation of therapy. So we think both in vitro and in vivo, the ability of these educated microglia to make CCL5 is dictated by NF-kappa B activation. Well, the last part I just want to talk about is closing the loop on how the T cell CCL4 production is induced, because the T cells don't normally make CCL5 just floating around in the bloodstream. They need to be told to make CCL4. And we hypothesized, based on evolving work that our lab is doing with Michel Manger's group, that maybe neurons were the missing link. And so we asked the question whether NF1 mutant neurons could induce T cell CCL4 production. And using both human-induced pluripotent stem cell central nervous system neurons and neurons from NF1 mutant mice, we're able to show that there is a paracrine factor made by NF1 mutant neurons that induces naive T cells to make CCL4. That's shown here. Here are the conditioned media from 2 mutants versus the control, and you can see higher induction of T cell CCL4 with just the neuron conditioned media. What was in that neuron conditioned media, both by a proteomic discovery and an RNA sequencing approach, was midkine, a member of the pleiotrophin family, which is known to be important for neuronal function, but hadn't been previously implicated in T cell activation. If you add midkine by itself, you can activate the T cells, and in here you can measure the amount of midkine that is made by the NF1 mutant neurons. And then lastly, just to show that this is truly midkine in the conditioned media, if we add neutralizing antibodies against midkine, the effect of the neuron conditioned media is now attenuated. NF1 mutant retinal ganglion cells are the source of the midkine because they're the cells whose axons project into the optic nerve. And just examining these optic glioma mice, you can see that there are many more midkine expressing cells. That's also true for NF1 associated pilocytic astrocytomas that have NF1 mutant neurons in them relative to either control brain or non-NF1 pilocytic astrocytomas. So the way that we envision things in this neuron, immune, and the cancer cell circuitry is that NF1 mutation in the neurons leads to an elaboration of midkine, which activates the T cells to make CCL4, and that T cell produced TCL4 now stimulates microglia to make CCL5 and promote the growth of the tumor. And just to show you, here's midkine activation of the T cells. And if you take the T cell conditioned media from these T cells and you add them to microglia, they now make CCL5. So this is the circuit as best as we have dissected it so far. Neurons making midkine that act on T cells to induce CCL4 production. So the idea that T cells can regulate optic glioma growth allows us to put these cells front and center in glioma pathogenesis, and raises a lot of questions about what other neuronal states might activate T cells, what additional T cell populations dictate development and progression, and what other mechanisms do T cells employ to establish a permissive microenvironment. It also suggests that T cells could be transducers of systemic disease. And one of the unexplained systemic diseases involving T cells is asthma, where patients with NF1 and sporadic tumors who harbor asthma have a twofold reduced incidence of brain tumors. And this is something that Jit Chatterjee in the lab is currently exploring using our optic glioma mice. It also suggests that T cells could be natural regulators of tumor growth and that we may be able to hijack the body's own immune system by teaching the T cells to no longer participate in this regulatory pathway. And then last, it also suggests that neurons create set points for cancer penetrance and progression, an idea that's been at the basis for the field of cancer neuroscience, and suggests that a number of insults, whether it's a hyperexcitable state, an injury, or a developmental disorder, may impact on the ability of neurons to participate in the genesis of tumors, as well as suggests new repurposing opportunities for therapeutic agents. To learn more about the role of neurons, I highly encourage you to attend Yuan Pan's talk tomorrow at 2:45 on NF1 mutation driving neuronal activity-dependent optic glioma initiation. It will be fabulous. So I want to stop there and thank the members of my group, and then thank the folks that I've had the good fortune to collaborate with, in particular, Michel Manger, funding agencies, and most importantly, our patients and their families.
So David, I think you can keep going because we will have the questions at the end.
Sounds perfect.
So,
well, it is my absolute pleasure to introduce Dr. Stephen Rhodes from Indiana University, who's going to speak on the immunology of peripheral nerve sheath tumor microenvironment. Stephen, welcome.
Thank you to the meeting organizers for the opportunity to speak today. I have no conflicts to disclose. I show this introductory slide just to provide a broader perspective on just how far we've come over the past decade in the treatment of NF1-associated plexiform neurofibroma. Looking back just over 10 years ago, we had essentially no effective medical therapies to treat patients with plexiform neurofibroma. And fast forward to the present day, 2021, we now have multiple drugs with established efficacy in phase 2 trials, and one drug, selumetinib, a MEK inhibitor, which was just approved this past year by the FDA for the treatment of plexiform neurofibroma in children with NF1. And so I think a key question is, how did we get here? And unequivocally, I would contend that the development of preclinical genetically engineered models that recapitulate many facets of this disease with a high degree of fidelity have proven themselves to be instrumental in catalyzing clinical translation over the past decade. In many ways, these genetically engineered mouse models have provided an unparalleled window of insight into the complex cellular interactions that exist between tumorigenic Schwann cell precursors and multiple other lineages, including stromal elements, vasculature, and infiltrating immune cells, which comprise the tumor microenvironment. And so this slide is to summarize several decades of work that has been done by multiple laboratories, just a subset of which are shown here, that has led us to the collective realization that it's not just the NF1 deficient Schwann cell that exists in isolation, but rather there are multiple cell lineages and immune effector cells within the tumor microenvironment that have properties to dictate both tumor initiation and tumor progression. Yet despite the incredible progress that's been made, I think in many ways our understanding of the immunologic complexities of these tumors remains in its infancy. And so over recent years, new technologies have emerged that are allowing us to characterize in unparalleled detail the immune landscape of these tumors. One approach we found particularly informative has been the use of RNA sequencing coupled with multiplexed inhibitor bead mass spectrometry, or MIB-MS for short, which uses beads covalently bound to multiple ATP competitive type 1 kinase inhibitors that can pull down functionally activated kinases from tumor lysate, which are then quantified based on their peptide signature by mass spectrometry. And this technology is able to capture not just the expression, but the functional activation state of upwards of 80 to 90% of the expressed kinome. And so we've collaborated closely with Dr. Steve Angus and Gary Johnson to apply these methodologies to understand the kinase signaling paradigms that are operant within the neurofibroma microenvironment. We've also sought to couple these technologies with advances in single-cell transcriptomics, whereby we can begin to construct a cellular atlas of these tumors. And finally, the use of digital spatial profiling on native formalin-fixed paraffin-embedded tissues that has enabled us to unlock archived specimens from human patients longitudinally across various stages of the neurofibroma to MPNST continuum, where we can quantify using molecularly barcoded probes the expression of key signatures of immune-related genes, signaling pathways, and cellular subsets. And so we've begun to use MIB-MS to characterize in a broad and systematic fashion the landscape of activated protein kinases within the plexiform neurofibroma microenvironment. And you can see here from the kinome tree and the volcano plot in the upper right-hand corner that there are a number of activated kinases with putative roles within multiple cell lineages, including AXL, the CSF1 receptor, which is critical for the recruitment of macrophages that we know heavily infiltrate these tumors, SRC, RIPK3, FES, which collectively modulate the biology of multiple immune cell lineages, as well as cyclin-dependent kinases CDK4 and CDK6, which are important both in the proliferation of Schwann cells as well as in other non-neoplastic lineages within the tumor milieu. So coupling these data with RNA sequencing, we can plot out orthogonally the log-fold changes in MIB binding on the x-axis and transcript expression on the y-axis, which shows several of these kinases to be both highly overexpressed at the transcriptional level, as well as demonstrating increased ATP competitive bead binding that we've subsequently validated by Western blot using phosphoantibodies. We've also shown in our recently published work that the loss of neurofibromin invokes a signature of cellular senescence in tumorigenic Schwann cells, and that following a transient burst of proliferation, these cells ultimately undergo growth arrest and upregulation of senescence-associated beta-galactosidase, as well as multiple components of the INK4ARF tumor suppressor locus that is well known as a master regulator not only of senescence, but also a phenomenon known as the senescence-associated secretory phenotype, which triggers cells to produce a number of highly inflammatory cytokines. And so one area that we're actively investigating is the potential relationship between cellular senescence and the pro-inflammatory phenotype in plexiform neurofibroma, and how these senescent cells may influence the infiltration of other inflammatory immune lineages within the tumor microenvironment. Work in these genetically engineered mouse models has also informed clinical translation of multiple therapeutic agents in the treatment of plexiform neurofibromas. Shown here to highlight seminal work that has been done by Brigitte Wiedemann, Andrea Gross, Eva Dombey, and colleagues at the NCI that ultimately led to the FDA approval of selumetinib for the treatment of plexiform neurofibromas in children, but also to point out a number of ongoing trials with other MEK inhibitors, binimetinib, trametinib, and mirdametinib, which are also showing encouraging preliminary results. And finally, other strategies targeting kinases that are upstream of RAS, including cabozantinib, which together with our colleagues Michael Fisher, Chi-Shen Shi, Jayshree Blakely, and others in the Department of Defense, NF Clinical Consortium was just recently published, as well as work some years previously with imatinib, led by Dr. Kent Robertson. And so we've been applying these new technologies to more deeply define the pharmacologic signatures of therapeutic agents, which have demonstrated efficacy in the clinic. And in so doing, this has allowed us to achieve new insights, not only into the pathobiology of these tumors, but also to begin to illuminate a novel drug combinations with efficacy in preclinical models that we hope will serve to inform further early phase trials and clinical translation in this space. So when we perform RNA sequencing of murine plexiform neurofibroma treated with MEK inhibitor, we find that this impacts not only the tumor-initiating Schwann cells themselves, but there's also actually quite a pronounced repression of immune cell trafficking and inflammatory signatures, as shown by the blue tiles that predominate on this heat map, as well as a number of immune-related kinases that appear to be reflective of a broader change in the infiltration of inflammatory immune cells into the tumor microenvironment, which we've subsequently confirmed is indeed the case at the histological level. Together with our colleagues, Michael Fisher, Jayshree Blakely, and the Department of Defense NF Clinical Consortium, we also recently conducted a mechanistically linked preclinical and multi-center phase 2 trial of cabozantinib in adults with NF1-associated plexiform neurofibroma, which showed a 42% partial response rate. In parallel studies, kinome analysis of plexiform neurofibroma tumor tissue from mice treated with cabozantinib revealed reductions in a number of kinases, which were actually unanticipated to us at the time, including DDR1 and DDR2, which are collagen binding kinases, as well as AXL, MERTK, and MET that have collectively diverse roles in tumor immunology, angiogenesis, and other processes as well. When we further perform RNA sequencing of murine plexiform neurofibroma and utilize a technique called enrichment mapping to help us ascribe biological meaning to these complex data sets in a visual way, with each node representing a signature of genes, we can begin to appreciate the major biological themes and signatures that comprise these tumors. And so, given the complementary target spectrum and cell lineages impacted by selumetinib, which blocks not only RAS-MAP kinase signaling, but also the proliferation of neoplastic Schwann cells, and importantly, as we now recognize immune and inflammatory responses within the tumor microenvironment, together with cabozantinib, which we now recognize predominantly targets the stromal microenvironment and neoangiogenesis, we reasoned that there could, in fact, be utility for combining these agents in the treatment of plexiform neurofibroma. And we found indeed that when we treated mice at low doses of selumetinib, 25% of the standard dose, and cabozantinib at 50% of the standard dose, we do in fact observe an additive effect of combining both of these agents together. And so these findings are now being moved forward into a multi-center phase 2 trial by our colleagues, Carlos Romo, Jayshree Blakely, Michael Fisher, and Brigitte Wiedmann, with the goal of testing whether combining both of these drugs at lower doses might be able to help us achieve deeper therapeutic responses in patients than with either agent alone, while hopefully minimizing toxicities. I'd like to transition now to discuss the progression of plexiform neurofibroma to atypical neurofibroma and ultimately malignant peripheral nerve sheath tumor, which is the leading cause of premature death in persons with NF1. We've recognized, based on work from Dr. Eric Legis, Doug Stewart at the NCI and colleagues, as well as other groups, that a very high proportion of human atypical neurofibroma and MPNST exhibit copy number loss of the CDKN2AB, or INC4ARF tumor suppressor locus. And so, informed by these insights, our laboratory generated genetically engineered mouse models which demonstrate a causal and gene dosage dependent link between ablation of the CDKN2A alternate reading frame, ARF, and the development of both atypical neurofibroma and MPNST. And this has also been subsequently shown by Dr. Nancy Ratner's lab as well. And so we've begun to leverage these models where there is loss not only of CDKN2A and its alternate reading frame, but also CDKN2B, which is frequently co-deleted with CDKN2A in the majority of human NF1 patients with atypical neurofibroma and MPNST. And we're beginning to use these models to develop a cellular atlas of these tumors across multiple stages of the neurofibroma to MPNST continuum, and beginning to understand both reciprocal as well as unique features that are germane to individual components of this key tumor suppressor locus that we believe may have important therapeutic implications. These are some preliminary data from mice with combined inactivation of NF1, CDKN2A, and B in embryonic neural crest driven by periostin Cree, in which we've been able to catalog the transcriptomes of about 60,000 cells. And using a dimensionality reduction technique called uniform manifold approximation, or UMAP, to project these data onto a two-dimensional plane and then resolve various populations of immune subsets, T cells, natural killer cells, mast cells, macrophages, neutrophils, and myeloid-derived suppressor cells, along with stromal components, tumor-associated fibroblasts, endothelial cells, as well as tumorigenic cells, both mature Schwann cells, as well as cells that express a stem-like phenotype characterized by the expression of nestin and other primitive neural crest markers, which our colleagues, Dr. Luis Parada and Lu Lei, have demonstrated are pivotal to the initiation and propagation of these tumors. And so coupling this technology with MIB-MS data from bulk tissues, we've begun to chart the expression of some of the most highly activated protein kinases at single cell resolution within tumor, stromal, and immune lineage subsets. And we can see kinases such as AXL, CDK4, and CDK6, and others that have heterogeneous expression across the cellular landscape. Some that predominate in stromal and tumor-initiating cells, and then others that predominate in immune cell subsets, infiltrating macrophages, neutrophils, and other lineages. And so collectively, we're hopeful that this approach will provide value not only in deepening our fundamental understanding of the biology of peripheral nerve sheath tumors in NF1, but also in understanding what cellular components within the tumor we are actually impacting when we administer these therapeutic agents in vivo. One of the major limitations that we face clinically is that malignant transformation of plexiform and atypical neurofibroma often occurs with little or no warning. And presently, aside from genomic loss of CDKN2A/B, we don't yet have the ability to molecularly stratify which tumors are at highest risk of undergoing malignant transformation. Reliance on obtaining fresh or high quality flash frozen tissues is a major challenge. And further complicating matters is the fact that various stages of tumor histology, ranging from plexiform neurofibroma and atypical neurofibroma all the way to MPNST, can often coexist even within the same biopsy specimen. And so to overcome these limitations, we extracted RNA and performed a spatial gene expression profiling from restricted regions of tumor that were carefully annotated by our collaborator, Dr. Andrew Horvath, in the Department of Pathology at UCSF. And in this pilot study, we profiled 35 NF1-associated peripheral nerve sheath tumor specimens from subjects who had a range of multiple tumor subtypes across the neurofibroma to MPNST continuum. We saw that the three major tumor subtypes, plexiform, atypical neurofibroma, and MPNST, broadly separated based on their global gene expression profiles, as demonstrated by this principal component plot. Further, when we looked at the expression of signatures of genes that are relevant to various biological pathways, as shown on the left, the tumors separated into three distinct molecular subclusters, and our attention was drawn to cluster 3, shown here at the right, which contained the MPNST, but also, surprisingly, there were several benign tumors with either plexiform or atypical neurofibroma histology that nonetheless had molecular signatures that were quite similar to that of MPNST. And so we wondered whether this pattern of gene expression might be predictive of neurofibromas that are at high risk of undergoing malignant transformation. And intriguingly, 100% of the subjects with benign tumors in this molecular subcluster developed MPNST within a relatively short window of time of 2 years or less, as compared to relatively fewer in the other subclusters. And so while there are clear limitations to this being a small, retrospective study with subjects that were deliberately selected because they had multiple tumors across the neurofibroma to MPNST continuum, nonetheless, we think this is a key first step in beginning to develop molecular or even immunologic diagnostic signatures that could be used to identify neurofibromas at high risk of undergoing malignant transformation. And so we're in the process of conducting further retrospective analysis to validate this, as well as a larger prospective study where we hope such an approach could ultimately guide early clinical intervention in patients who are deemed to be at high risk of developing MPNST. And so with that, I'd just like to thank Dr. Wade Clapp for his outstanding mentorship during my fellowship training, and the many members of the Clapp Lab, Dr. Steve Angus and the Angus Lab, who were instrumental in performing this work, as well as a myriad of other collaborators, both internal and external to Indiana University, in particular, Jayshree Blakely and Mike Ferguson, who have been clinical mentors to me through the Francis Collins Scholars Program funded by NTAP, as well as other grant support from the K-12 Pediatric Physician Scientist Development Program, the Green Scholars Program, the Department of Defense, the Precision Health Initiative at the Indiana University School of Medicine, and finally a developmental and hyperactive RAS tumor spore led by Drs. Wade Clapp and Kevin Shannon. And so thank you again, and would be happy to take any questions at this time.
Okay, thank you, Stephen. As we were saying, we will have a question answer session at the end of the 4 talks. And now it's my great pleasure to introduce the next invited speaker, whom I personally thank for having accepted our invitation, who is Dr. David Reardon, who directs the Neuro-Oncology Center at Dana-Farber and Harvard Medical School. Dr. Reardon, together with his colleagues, has really done incredible, exciting work recently to characterize the tumor microenvironment of glioblastoma. And I think most importantly, show to all of us how this information can be used in real time for decision-making process in the clinic. The title of David's talk is State of Immune System in Glioblastoma Implications for Immunotherapy Trial. David, the virtual podium is all yours.
Well, thank you very much. Antonio, can you all hear me okay?
Yes, everything okay.
Okay, great. I'll advance the slides here. I'd like to thank the organizers for the opportunity to present at this conference. And I do preface that the talk is focused on glioblastoma, but I think a lot of what I'll be highlighting could be extrapolated or useful for other CNS tumor types going forward. Okay, good. All right, so just a quick snapshot for those who are not familiar in medical oncology. Immunotherapy, in particular immune checkpoint drugs, have transformed the treatment of many different types of cancers now. This is probably obsolete, even though I only made it a month ago, of 49 different FDA-approved indications for these immune checkpoint drugs that have evolved since 2010. Unfortunately, we don't see glioblastoma or other primary tumors on the list, but these agents are now routinely being used for many, many different types of cancers in medical oncology. Unfortunately, in neuro-oncology, we have not had such great experience. And here's a slide summarizing some of the phase 3 studies, unfortunately, all of which have been negative, looking at a variety of different immunotherapy approaches for glioblastoma, including 3 potentially registration studies for with Opdivo, a PD-1 targeting checkpoint inhibitor. So, interestingly, that's the story for GBM, but these drugs do seem to work in CNS metastases. And this is data from a recent publication of combining PD-1 with CTLA-4 for CNS mets in both asymptomatic and symptomatic patients, showing pretty remarkable rates of durable disease control in the CNS in this disease. So what is unique and different about glioblastoma and maybe some of the other primary tumors in the spectrum of glial tumors in the central nervous system? We've heard some of the elegant work from Dr. Gutman and his group highlighting some of these factors. Unfortunately, I think of it as kind of a perfect storm where there are a number of different factors that stack the deck, if you will, against immunotherapy in this disease. We know the glioblastoma and other glial tumors are tumors of low inherent immunogenicity. They tend to have a low mutational burden and low PD-L1 expression. And they tend to secrete a variety of different factors and molecules that lead to a dominantly suppressive microenvironment within the tumor that attract myeloid cells from the bone marrow and transform those that are intrinsic to the CNS, the microglia, to be very suppressive. We know from Peter Fecchi's lab at Duke that there is some sequestration in the bone marrow of T cells, and then there is an aggregate of very hostile environmental conditions within high-grade gliomas, in particular hypoxia, low nutrient availability, as well as acidosis that are very detrimental for T cell vitality and function. And then importantly, other considerations, and I want to come back to this in the story of corticosteroids that we tend to utilize in our neuro-oncology patients. This is a very nice multiplex immunofluorescence that Amy Heimberger, who's now at Northwestern, was kind enough to lend to me, highlighting the paucity of T cells within malignant glioma tumors. Those are highlighted in either red or fuchsia colored, and you can see the overwhelming predominance of myeloid cells, the CD68 yellow cells here, totally outnumbering and outgunning any T cells that made their way into this microenvironment. And as mentioned, these macrophages are phenotypically very immunosuppressive and tumor supportive. So what can we do to overcome that T cell desert, if you will, in these tumors? There are a variety of different strategies. I think some of the exciting work that's now being done with oncolytic viruses, genetically engineered viruses that are injected into the tumors, have generated robust immune responses in the tumor microenvironment. We've been doing some work with another strategy of utilizing a personalized cancer vaccine approach to try to sensitize systemically the immune system and then drive these cells into the tumor microenvironment. And the pipeline for this here at the Dana-Farber was led by my colleague, Dr. Kathy Wu. Gavin Dunn at WashU, Dr. Gutman's colleague, has something very similar ongoing at WashU for patients. But basically, we compare tumor and normal DNA, RNA expression, identify the coding mutations that are unique to the individual patient's tumor, predict those that are most likely to be immunogenic for that individual patient, and then synthesize, in our case, up to 20 to 30 amino acid long peptides corresponding to these coding mutations. And we refer to this as neovax at our institution. And the first study with this approach, highlighted in the upper left corner, led by my colleague in melanoma, Patrick Ott, where the vaccine, 20 different peptides corresponding to coding mutations generated per patient and administered along with poly-ICLC using this 5 priming dose followed by 2 booster dose schedules. And in their hands in the melanoma study, seeing very nice CD4 and CD8 responses that were only detectable after vaccination. They were specific to the vaccinated neoepitope peptide, not cross-reacting to the wild-type peptide, and many of them were polyfunctional. Impressively as well, looking at recent follow-up of some of these treated patients who are still alive and doing well, you can see the ELISpot reactivity against vaccinated neoepitope peptides for some of these patients being quite robust now 4 years later, even though their vaccine ended after 20 weeks initially of treatment. One of the things that's emerged is that these vaccines can induce immunogenic cell death. These T cell responses associated with the vaccine can induce immunogenic cell death, which can lead to epitope spreading, where the immune response can broaden not only against the vaccinated neoepitope peptides, but other neoepitope peptides that were not included in the vaccination, but identified in this case in the upper panel for a study in melanoma patients, and seeing some of these non-vaccinated neoepitope peptide responses early on and also out to 4 years later. And in a study of a number of different solid tumors in the adult sector where this neoantigen vaccine approach was applied, those who did have similar evidence of epitope spreading had significantly improved progression-free survival relative to those who did not have evidence of epitope spreading. So in glioblastoma, it's a very different situation than melanoma, as we've talked about, a much less immunogenic tumor here. So we next turned from melanoma to a less immunogenic tumor here, looking at the application of this approach in glioblastoma. We have a fairly handy window when patients are recovering from surgery and undergoing radiation therapy to generate their personalized vaccine. It does take about 3 months to do this per patient, although the timing is coming down. And then, similar to the melanoma study, giving this priming boost schedule of personalized neoepitope peptides along with poly-ICLC as the adjuvant. Like the melanoma study we published and showed in glioblastoma, in some patients we were able to generate robust de novo polyfunctional T cell responses against the mutant but not the wild-type peptides. But interestingly, patients who responded were only the patients who responded were those who were not on dexamethasone. Patients who were on dexamethasone during antigen priming completely failed to respond to the vaccine. We were able to demonstrate in patients who had tumor resected after their vaccination at the time of evidence of progression on MRI to determine if those progressive findings were pseudo-progression or real progression, that in the patients who responded systemically, those who were not on steroids, like patient 7 and 8 here, we did see a robust infiltration of T cells into the tumor. In contrast, patient 11 is an example of a patient who was on steroids, did not respond systemically, and we did not see any T cell influx into the tumor. And finally, we were able to demonstrate that the vaccinated neoepitope peptides not only generated T cell responses systemically, but we were able to isolate T cells post-vaccination and confirm through some fairly elegant work by my colleagues that the T cells were isolated from the tumor post-vaccination were indeed specific to the vaccinated neoepitope peptide. So our next steps with this approach it makes sense to combine this with PD-1 as a way to further enhance those anti-tumor T cell responses in the tumor microenvironment. But we're wanting to do this in a careful and cautious way and look at the potential implications of timing. We know when our immune system responds naturally to an infection or other foreign entity, it happens in a very carefully orchestrated sequential pattern. Everything doesn't happen simply at once. So in thinking about combining checkpoint blockade with antigen priming in a vaccine strategy, we wondered if there could be a difference in how PD-1 is timed relative to antigen priming. And there is some emerging data to suggest there could be an impact on timing. One of the sources of this data comes from Arlene Sharp, who's the head of immunology at Harvard Medical School, one of our colleagues here. And in infectious disease models, she recently, in her lab elegantly, demonstrated that in the PD-1, PD-L1 knockout mice, they tend to have a poor memory response to influenza and tend to be sicker when they're re-exposed to influenza, indicating that PD-1 may be having some impact on their ability to respond immunologically. Samir Khalif at this recent publication as well demonstrated that there is an impact, can be an impact on timing. And in this paper, 2 different PD-1 resistant cancer models were employed, looking at PD-1 or vaccine alone had no impact on tumor growth nor any impact on survival. When combining PD-1 with vaccine, timing did matter in this experiment. If the PD-1 was given 7 days prior to starting vaccination, which is the darker green line here, it made no difference. The mice had no benefit. However, if PD-1 was given essentially at the same time or immediately after vaccination, not being blocked prior to antigen priming, it made a significant difference in both models in terms of tumor growth as well as survival. And in this paper, they did a lot of elegant work to identify the mechanism associated with this. And I bring to your attention at the bottom panel here, the CD8 responding T cells, antigen-specific responding T cells in the mice who had PD-1 prior to their antigen priming. These T cells were exhausted, manifest by a dual expression of PD-1 and CD38, as opposed to a lower level of these exhausted cells, a non-significantly elevated level if the PD-1 was given after vaccination. So in our study that's ongoing now that we are enrolling patients to, we're using the exact same schedule of vaccination as we had before, but we're introducing PD-1 blockade with pembrolizumab before, during, or after antigen priming, and are going to do a deep dive into the immune responses to see if what has been brought to attention in mouse models may be relevant in human patients. I want to shift now and come back to this issue in clinical development for neuro-oncology, thinking about corticosteroids, and is this a real problem or not for immunotherapy approaches for our patients. I already mentioned the many factors that these tumors are exploiting to generate a suppressive microenvironment, but are we adding to that and contributing to that with the use of corticosteroids? And although there has been some data in the literature, we undertook a fairly detailed analysis in our mouse models of glioblastoma, syngeneic mouse models of glioblastoma, to look at this, and published it recently in Clinical Cancer Research. And this was work led by one of our junior pathology faculty, Brian Irogaluscu. And in this highly immunogenic, very PD-1 responsive syngeneic GBM model, GL261, we had previously published, and many others have confirmed, a high rate of response and benefit with PD-1 therapy. Here you can see we cured 80% of the mice with treatment starting 6 days after tumor implantation, and the controls typically dying by somewhere around 30 days. When we gave increasing doses of dexamethasone along with the PD-1, we had a progressive worsening of survival and outcome. When we combined PD-1 with radiation in both the 261 as well as the CT2A syngeneic models, we did see enhanced benefit of combining PD-1 and radiation therapy, but that enhanced benefit was completely lost when we added dexamethasone in these treated mice in both models. Looking mechanistically, we were able to identify significant decreases both in the tumor microenvironment as well as draining cervical lymph nodes, the spleen, and the thymus of CD4, CD8 T cells. And here's just a multiplex immunofluorescence showing a graphic picture of that in the control versus the dexamethasone treated mice. And when we looked functionally and separated out these T cells, we could see significantly impaired gamma interferon secretion associated with the use of dexamethasone therapy, as well as an induction of apoptosis markers indicating dexamethasone decreases quantitatively by inducing apoptosis, as well as impairs the function of T cells. We then turned to the clinic, and to look at our clinical experience, we have a database now of a couple hundred consecutively treated GBM adult patients, IDH wild type treated with PD-1 therapy on clinical trial or compassionate use. And when we looked at the analysis of outcome of these patients, 181 patients, the use of dexamethasone versus no dexamethasone was the most important, other than MGMT status, predictor of overall survival of these patients, with a worsening of survival associated with higher dexamethasone doses. And here's the Kaplan-Meier curves identifying the non-dex receiving patients in the solid line, and then the dexamethasone treated by dose in the dotted lines. And this is a Kaplan-Meier just highlighting the same outcome, but looking at it from a multivariate adjustment, looking for other relevant contributing prognostic factors. That still the use of dexamethasone identifying the patients as having a poorer outcome. So I think this highlights something very important that we need to keep in mind as we move forward with immunotherapy for our treatment. There are a lot of hurdles and challenges to overcome. We should try to avoid if at all possible. And I know it's challenging because there are not a lot of good options in patients when they do develop symptomatic cerebral edema, but try to limit as much as possible the concurrent use of corticosteroids. The last story I want to highlight is a discovery story that we embarked on when we learned and saw the disappointing results of immune checkpoint inhibitor trials in glioblastoma patients. It begs the question of, well, what are we missing, and why are these tumors so different relevant to other tumors that are responding well? So this is work on a project I initially talked with Mario Suva from the MGH about, who's done a lot of elegant work identifying single-cell RNA-seq in tumor cell populations. And in collaboration with Kai Wucherpfennig, who oversees the immuno-oncology group here at the Dana-Farber, and a very talented grad student in Kai's lab, Nathan Matthewson. And what we did initially was simply ask the question, what do we not know, and how can we try to get an answer or get some insight into that question? And we simply took 31 GBM tumors, some of them IDH wild type, some of them mutant, some of them on dexamethasone, some of them not, some of them newly diagnosed and recurrent. We basically took what we could directly from the operating room, brought it up to Kai's lab, and he separated out the CD45 negative cells and the CD45 positive cells. The positive cells we then separated into immune cell subsets, and then with Mario's help performed single cell RNA-seq. And what we were able to identify on the immune cell populations, first of all, we validated that the patients who are on steroids versus not on steroids had significantly lower numbers of T cells isolated from the tumors, as you can see highlighted here. In separating out the T cells, we broke them up into 5 main components, and interestingly saw no difference in the distribution of those cell subsets based on the IDH status of the tumors. We identified 6 kind of profiles of CD4 and CD8 T cells from these isolated tumors, and each profile is highlighted in the linearly here. And within each profile are the IDH wild type and negative tumors side by side. You can see the different profiles that were identified here amongst these T cells. And one of the things that stood out quite surprisingly early on in this evaluation was the presence of upregulated natural killer cell related genes. These hadn't been reported as being upregulated on T cells or tumor infiltrating T cells previously, but we noted a few of them. In particular, we focused on CD161, which, as you can see in the blue bars, quite upregulated in both CD8 and CD4 T cells isolated from the tumors by flow cytometry. We looked at the T cells that were clonally expanded within the tumors, thinking those were the cells that were most likely reactive to tumor-associated antigens. And in these clonally expanded T cell subsets, again, we saw a striking upregulation of some of these natural killer cell markers, including KLRB1 highlighted in the green bars here. So we went back to the literature and asked, what do we know about KLRB1? And we know it's been known to be an important inhibitory molecule receptor associated with NK cell cytotoxicity when it binds to a ligand called CLEC2D, which is also known as lectin-like transcript 1 or LLT1. Interestingly, Michael Weller and Patrick Roth from Zurich had published a few years ago that CLEC2D is known to be upregulated in glial tumors, and its expression level increases with increasing grade of glial tumors. We went to our tumors, went back and looked at CLEC2D expression, and did indeed confirm that it's upregulated in both the tumors as well as the myeloid cells within the tumors. We identified its expression within a variety of different glioblastoma cell lines. And here's some of the RNA in situ hybridization confirming tumor expression. This is just a graphic to show that the KLRB1 molecule, its upregulation in the TILs isolated from the tumors we analyzed. And actually, here you can see significantly higher rates of KLRB1 relative to PD1 on the CD4 and CD8 T cells isolated from these tumors. We then went and I don't have much time to go into detail here, but in vitro studies confirming that this KLRB1 does contribute to T cell inhibition and inactivation in vitro studies using Cas9 inactivation as well as blocking antibodies. And then in vivo confirmed in humanized mouse model overexpressing NY-ESO, that when we treated these tumor-bearing mice with CARs specific for NY-ESO that either had intact or inactivated KLRB1, the mice who were treated with CARs that had inactivated KLRB1 had significantly higher survival, indicating we were blocking a potentially relevant, important immunosuppressive molecule in these tumors. Finally, we went and looked to see if this was simply a glioblastoma unique immunoinhibitory circuit of KLRB1 binding to tumor-expressed CLEC2D. But basically in every tumor we've analyzed, we've discovered and noted that KLRB1 is upregulated within the tumor-isolated lymphocytes. In the upper panel is melanoma, liver cancer in the bottom left, lung cancer in the upper right, and colorectal in the bottom right. And I highlight in the middle bottom of each of these cancers is the PD1 Tisney plot, and KLRB1 is immediately to the left of that. In melanoma, there's more PD1 than KLRB1, but there's still significant KLRB1 expression. And in the other tumors, there's at least as much KLRB1, if not more, than PD-1 expression in these tumors, indicating that there is another relevant immunoinhibitory circuit that certainly may be inhibiting anti-tumor immune responses and immunotherapy in glioblastoma that may be relevant in other tumors. And we're currently generating humanized anti-CD1, 61 or KLRB1 antibodies as a strategy to block this immunoinhibitory circuit in glioblastoma and other cancers. So to conclude, although immunotherapy, and particularly immune checkpoint therapy, is now a cornerstone of many different cancers, unfortunately, it has not been effective in our patient population. This is a tumor type. Our glioma tumors are identified and classified now as immune refractory based on the lack of benefit from single agent immune checkpoint therapy. There are a number of strategies, though, that we can utilize where we can convert this cold tumor microenvironment and suppressive immunomicroenvironment to a more inflamed immune supportive microenvironment. And in our hands, we're looking at neoantigen vaccination and carefully trying to identify if there's a role of timing relative to PD-1 blockade. I think in our field, unique to other cancer indications, we have to be very careful about dexamethasone therapy. We all know it has lots of side effects for patients, but in particular with immunotherapy and anti-tumor immune responses, it may be very potently detrimental. And finally, I think that we still have a lot to learn about what's driving immunosuppression and the inability of generating robust anti-tumor immune responses. And we need to do more discovery. We need to better understand the different components of the tumor microenvironment, including the dominating myeloid component, and our work highlighting one previously unrecognized immunoinhibitory circuit of KLRB1 and CLEC2D is just one example of what we still need to learn going forward. So with that, I will close, acknowledge the many members of our team that I have the privilege of working with every day, some of the key contributors to the work highlighted by the red bars, and highlight some of the funding support that made all of this, all of our work possible. So thank you very much again for the opportunity to contribute.
Thank you, Dr. Reardon. That was a spectacular talk, covering a tremendous amount of landscape in our understanding of brain tumors. So thank you for sharing your observations with us. Our final speaker was chosen as a platform speaker. That's Dr. Harish Vasudevan, and I'm sure I butchered that completely. So we'll just we'll have him introduce the correct pronunciation of his name. Harish is from the University of California in San Francisco, and his talk is entitled Epigenetic Reprogramming Drives Schwann Cell Transformation and Treatment Resistance in Malignant Peripheral Nerve Sheath Tumors. Welcome, Harish.
Thank you, Dr. Good afternoon. Thank you for joining us. My name is Harish Vasudevan. I'm one of the Holman Pathway Research Fellows in the Department of Radiation Oncology at UCSF. And today I have the pleasure of sharing our work looking at how epigenetic reprogramming drives Schwann cell transformation and treatment resistance. What I have here is the famous Waddingtonian epigenetic landscape. And if there's one thing I can communicate today, it's that where a cell sits in this landscape affects not only transformation, but also how our patients respond to therapies. What I have here in this Waddingtonian representation is a pluripotent stem cell in white, differentiating along the Schwann lineage in purple. NF2 loss leads to development of schwannomas in green. NF1 loss leads to development of neurofibromas in blue. And what we really want to understand is how a cell moves along this plane upon transformation to MPNSTs. Stating with background, Schwann cells give rise to distinct tumor entities with different clinical behaviors in neurofibromatosis. Comes as no surprise or needs no introduction for this audience, but Schwann cells upon loss of NF1 can develop neurofibromas, which are shown here in blue. Upon loss of NF2, they can develop schwannomas, shown in green. While these tumors have very different molecular mechanisms of transformation, they do share some clinical attributes in that they're benign, do not metastasize, and respond to treatment. In contrast, malignant peripheral nerve sheath tumors, or MPNSTs, which can arise from neurofibromas or in rare cases from schwannomas, are malignant, metastasized, and are treatment resistant. If we look at outcomes from MPNSTs, this is specifically data from UCSF that we recently published. Both sporadic MPNSTs in black as well as NF1-associated MPNSTs in blue have a 5-year survival of about 60%. And so it's no surprise that MPNSTs are one of the leading causes of mortality in adult patients with NF1, which really motivates understanding. My outline for my talk today, I'll first share our multi-platform genomic analysis, which comprises methylation array profiling, RNA-seq, and whole exome sequencing of schwannomas, neurofibromas, and MPNSTs. I'll then discuss the consequences of PRC2 loss in tumor cell lines. PRC2, or the polyclonal repressive complex 2, is an important epigenetic regulator that has been well established to have a role in transformation to MPNST. Then I'll conclude by talking about how dedifferentiation might help inform mechanisms of MEK inhibitor resistance. No genomic analysis or talk would be complete without a clustering dendrogram. What I'm showing here is hierarchical clustering of our DNA methylation array data, which identifies 3 epigenetic subgroups of Schwann cell tumors. Cluster 1, which I've demarcated in red here, is almost entirely MPNSTs, which are NF1 mutated, CDKN2A deleted, and exhibit PRC2 mutations, either loss of SUS12 or EED, which are 2 core components of PRC2. In contrast, cluster 2, it comprises neurofibromas and MPNSTs. These are NF1 mutated. Many of them are CDKN2A deleted, but essentially none of them are PRC2 deleted. So these are PRC2 intact. Finally, cluster 3, shown in black, is almost entirely schwannomas. There are exactly 3 cases that are not, and all of those upon re-review appear to be Schwannian. These have a preponderance of NF2 mutations, but they're NF1, CDKN2A, and PRC2 intact. So this leads us to support the established model of Schwann transformation, which is NF1 loss leads to the development of a neurofibroma, and then additional hits, most notably CDKN2A loss or PRC2 loss, which leads to H3K27 trimethylation loss and aberrant epigenetic activation of certain genes, leads to MPNST transformation. And this has been supported in the literature now for a number of years. So that made us feel confident what we were finding was real. So to dig a little bit deeper, we performed differential methylation analysis between these clusters. And what we found is sites that were hypermethylated or silenced in cluster 1 were associated with transcription factors important for cell differentiation. In contrast, hypermethylated sites in cluster 2, as you might expect given the role of PRC2, was enriched for PRC2 targets, including the core components of PRC2, EZH2, and SUS12. If we look at the level of gene expression, what we find is that in cluster 2, the genes that are upregulated are associated with ectodermal or neural development. And indeed, if we look at the classic Schwann marker SOX10 or S100D, these are significantly decreased at the mRNA level, and cluster 1 is shown in red compared to cluster 2. Now, how does this look in cell lines where we can perform more mechanistic analysis? To get a sense for this, we collected a set of neurofibroma cell lines shown in blue, which are NF1 deficient, and MPNST cell lines shown in red, which have a smattering of NF1, CDKN2A, and SUS12 statuses. If we do RNA-seq expression of these cell lines, what we see is that the MPNST cells are enriched for upregulation of SUS12 targets, consistent with the idea of PRC2 loss being important. In contrast, the neurofibroma cells had increased expression of nervous system development genes, things like SOX10 and S100D. Now, these are obviously different cell lines. What happens if we actually go through and try to understand the effects of loss of PRC2? To do this, we integrated our RNA-seq data with published data 2 years ago in PNAS looking at the effects of PRC2 knockout on neurofibroma cells. And we find something striking, which is that the neurofibroma cell lines shown here in blue, or the neurofibroma cell lines with loss of just EZH1 or 2, which is insufficient to aggregate PRC2 activity, cluster as one set of tumors shown here. In contrast, the MPNST cell lines, as well as neurofibroma cell lines that are truly PRC2 deficient, so either combined EZH1 plus 2 loss or SUS12 loss, cluster separately. And if we look at the genes driving this difference, what we find is that transcription factors associated with dedifferentiation, such as FOXF1 and GLL1 SOX9, are increased in MPNST cells, as are classic RAS, MEK, ERK feedback regulators such as DUS6, SPRADI2, as well as the RAS target gene ETB4. Next, we wanted to see if these effects were direct or indirect. In order to do that, we examined published ChIP-seq data either in neurofibroma wild-type cells or neurofibroma cells with SUS12 knockout. Looking first at those dedifferentiation transcription factors in GRAILD1, SOX9, and FOXF1, we see that these promoters and gene bodies are highly methylated in the presence of SUS12. However, upon SUS12 knockout, there is essentially no methylation, and that correlates with upregulation at the mRNA level of these genes. So this suggests the dedifferentiation process is a direct chromatin effect. In contrast, we see something different for the RAF/MEK/ERG target genes, things like DUS6, SPRADI2, ETB4. There's very little methylation at these promoters and gene bodies at baseline, and therefore there's no methylation to be lost, and we don't see any differences upon SUS12 knockout, even though at the mRNA level, there does seem to be significant upregulation of these genes. So this suggests the RAF/MEK/ERG target genes are regulated in an indirect manner. Finally, looking at the effects of MEK inhibition in our NF1 deficient neurofibroma cell lines, we used the FDA-approved MEK inhibitor selumetinib, treated out to 10 days on NF1 deficient neurofibroma cells, and then analyzed these cells in order to understand mechanisms of intrinsic resistance. And what we find is this appears to be associated with decreased expression of the Schwann cell differentiation marker SOX10. So specifically, if we look at phosphoric, we see a nice inhibition at 1 hour, which rebounds at 1 day and then normalizes. In contrast, SOX10 levels, shown in blot form on the left and in blue quantified on this right plot, fall off. And we can recapitulate the SOX10 repression at the mRNA level. Finally, to understand these mechanisms of intrinsic resistance at single-cell resolution, we took NF1 deficient neurofibroma cells, treated them for either 1 or 2 days with selumetinib, and performed single-cell RNA-seq. What we identified were 8 cellular subpopulations across all 3 time points. So this UMAP is aggregating data from all 3 time points. And the 4 populations I want to draw your attention to, which change over time, are the differentiated Schwann cells, shown in yellow, the proliferating and undifferentiated Schwann cells, shown in teal, the proliferating and differentiated Schwann cells, shown in blue, and the resistant cells, shown in purple. Essentially, the differentiated cells go down over time, and the undifferentiated resistant cells go up. And that's exactly what's quantified here. Over time, that yellow differentiated population begins to go down, whereas the teal and purple undifferentiated populations go up. And if we look specifically at Schwann markers such as SOX10 S100B or proliferative markers such as Ki67 and TOP2A, we can see that this seems to form an axis of proliferation and differentiation in our single cell space. Finally, how does this correlate to PRC2? To ask this question, we took our PRC2 regulated signature and overlaid it on our single cell map. What we see is genes that are lost upon PRC2 knockout in neurofibroma cells are enriched in the differentiation clusters in this UMAP, while genes that are induced upon PRC2 loss are in the proliferative cluster. So to summarize, primary human MPNST tumor specimens demonstrate loss of PRC2 and Schwann cell differentiation. PRC2 loss in cell lines recapitulates this, and the effect on differentiation seems to be direct, while on MEK/ERG is indirect. And finally, MEK inhibitor resistance appears to arise at least in part due to resistance subpopulations associated with PRC2-mediated Schwann cell differentiation. So again, if I can leave you with one image, the Waddingtonian landscape for Schwann cell tumorigenesis, forming Schwannomas in green, neurofibromas in blue, and MPNSTs being pushed back up that differentiation hill due to PRC2 loss, which seems to be a partial regulator of why these tumors are resistant to MEK inhibition. With that, I'd like to thank my mentors, Frank McCormick, David Rowley, Adam Abate, and the folks in the labs who made this work possible, as well as my departments at UCSF. Thank you.
Okay, thank you, Harish. I think we can then use the next 7 minutes. I believe that's how much we have left for some, I am sure, exciting questions. Let me start you with a question to ask to my co-chair for that I compliment, obviously, for a very exciting talk. So the question we have from the audience is whether the neuron midkine production is electric activity dependent.
Right, so it's a great question, especially given the beautiful work from Michel Manger's group. In our hands, the increased neuronal activity represents an intersection between an NF1 mutation and the endogenous activity, and if we reduce the sort of baseline activity that's conferred by NF1 mutation to normal, then the midkine production is also downregulated. So in our hands, it does appear to be neuronal activity dependent, but reflects a mutation in the NF1 gene. Why don't I address one question to Dr. Rhodes? The individual asks, it appears that a higher percentage of plexiform neurofibroma transformed into MPNST is higher than the ANUBP in your evaluation. Any thoughts on that?
Yeah, that's a great question and a great comment. I think one of the limitations of the small cohort that we assembled, which again was highly selected, we selected for patients deliberately who had a broad spectrum of different tumors across the neurofibroma to MPNST continuum, and so by virtue of that, the percentage of MPNST in all three of the molecular subclusters is quite high. As to the difference between that second and first subcluster containing the most of the plexiform and the atypical neurofibromas respectively, I think one possibility, again, this is just a hypothesis, we'd have to go back and look at the data, but could be that it's quite possible that more of the patients, subjects who had atypical neurofibroma may have undergone not just biopsy, but complete resection of their tumor, just as is typically clinically indicated when that occurs. And so I'd have to go back and look at those data, but that's just one potential thought there, but an excellent comment and excellent question. Thank you.
Harish, do you have a sense of whether or not your data informs on the different cells of origin for schwannoma versus neurofibroma?
Yeah, it's a really good question. I think my data directly does not, but we've also done single cell RNA-seq of schwannomas and HA193, which is one of the common NF2 deficient schwannoma cell lines. And there does seem to be a different, quote, differentiation potential in terms of what those cells can become in vitro. Now, I'm a little embarrassed to give this particular talk in this session because everyone else had this beautiful in vivo data, while I, despite my mouse genetics love, did not leave the dish. But we are trying to do some of those experiments now with static nerve xenografts, both in nude and immunocompetent mice, to get a better idea of that.
Maybe one question to Dr. Reardon. In your experience with the GBM, is there an interplay between the CD4 and CD8 populations where they either cross-educate each other or manipulation of one impacts the other?
Well, I think in general, certainly having a robust CD4 infiltration within the tumor will be of benefit to the CD8 cells that are making their way into the tumor as well. Interestingly, in our vaccine platform, which I don't know if it's relevant or representative for some of the other kind of in situ vaccine approaches like the oncolytic viruses or other things that are being done. But we are seeing a significant number of CD4 reactive tumor antigen specific T cells within the cells, the TILs that are isolated, as well as the CD8. So I think we're beginning to better understand. And of course, Rob Schreiber has done a lot of elegant work from WashU as well, highlighting the benefit of the CD4 response for the cytotoxic CD8 response. But I think we still are learning a lot about that. But I'm gratified to see that we are seeing those CD4 TILs as well.
David, if I may, I would like to ask also a question to Dr. Reardon. It's about the myeloid microenvironment that obviously we always see as primarily immunosuppressive, but that so far, at least the early attempts to target this microenvironment hasn't been really very successful. But do you see this as really the next generation stage of immunotherapy, possibly in combination with vaccine and checkpoint inhibitors? Or on the other hand, do you see actually the myeloid microenvironment, the extent of macrophage infiltration, as potentially a window of opportunity? Because although we always think of this as an immunosuppressive environment, maybe there are abilities of myeloid cells to work together with T cells for a productive immune response. In other words, should we target with immunotherapies exactly those mesenchymal tumors that are showing the highest infiltration of both macrophages and T cells, in fact?
Yeah, I think that's a really extremely spot-on question, Antonio, and I think one that to me I think is going to make be the treatment that makes immunotherapy have an impact in these tumors is somehow being able to flip that myeloid component from being so dominantly suppressive to being immunosupportive. I don't know that we have to quantitatively deplete them. I think there were some initially strategies thinking about, well, let's just drive the numbers down, CSF1 inhibitors and things. And I don't think that necessarily is the answer. But I think if those molecules, the CSF1 inhibitors or other therapies, can simply flip a percentage of these myeloid cells, it may only need to be 5 or 10%. It doesn't have to be the whole thing. But if we can flip some of them to become more immunosupportive, I think it'll make I think that would be the deal breaker that would finally give immunotherapy a chance here. So I think you're absolutely right. The strategies we need to be focusing on, T cell strategies, obviously very critical and important. But if we don't address the elephant in the room, which is this dominating myeloid component within the tumor, those T cells aren't going to have a chance. And those T cells, those macrophages, if they're able to be more immunosupportive and have an anti-tumor effect, that in and of itself could be very exciting and very therapeutic.
But David, and then I will stop. It seems really puzzling to me that the T cell infiltrate usually, at least in GBM, correlate with the extent of macrophage infiltration. So, I mean, in other words, to have T cells, you need macrophages. And I don't know mechanistically how this works, and this may not be true in a low grade, but in GBM, this is really, I think, quite well established. So, I mean, either we believe that these T cells are completely corrupted, or maybe they are really there because they are doing something in terms of immune response.
I'm sorry. I think if there, yes, I fully agree. When we are seeing anti-tumor immune responses, there does seem to be compensatory suppressive factors that are triggered, upregulation of other immunosuppressive molecules in addition to PD-1, probably the KLRB1 we identified, but in addition, an enhanced activation of factors that are driving these myeloid cells, attracting them into the tumor, and I think making them more suppressive. So there does seem to be this compensatory reaction along with immune activation to suppress that and blunt that, particularly in the CNS with the inherent microglia we have, with the enhanced suppressive capability of these myeloid cells. So targeting enhanced T cell responses with somehow blunting this suppressive myeloid component, I think really is pretty critical.
Okay, probably we should now close the session as we are running out of time. David, maybe you want to say the last few words.
Well, I want to thank all of the speakers for presenting in what is a virtual world these days and being so accommodating. I want to thank Antonio for being my partner in crime and for hosting this virtual session of Hollywood Squares for Scientists. And look forward to seeing everybody in real life next year at the 2022 CTF meeting.
Let's look for that. Thank you, everybody.